All right, welcome back. Here is the last part of our hemoglobin journey, and we are going to tell you why we're doing all this. It's because hemoglobin is so deeply involved with an essential process, and it's so contained within the hemoglobin itself is the structural unit that causes most of these things to happen. Therefore, we can look at the hemoglobin itself, and biochemists can save the day on 3 different applications. And so we're going to talk about 3 of those. High altitude sickness, sickle cell anemia, and fetal hemoglobin, which is sort of tied up as sickle cell anemia, but because it's not involved with the disease state, it's a little bit different. So we'll talk about these 3 things that have medical applications where hemoglobin is involved with disease and health. So the first thing is something that everyone does. It's not a disease state at all, but it actually is a kind of sickness. When you go up to Denver, you take a plane flight to Denver and you're like gasping after walking down the block. At least I am. The reason for that is because literally you are at a lower partial pressure of oxygen. You are higher up from the Earth's surface. The oxygen is held down by gravity, and so you have a lower air pressure, less oxygen. Less air equals less oxygen. And so the thing that you might notice is if you have a friend in Denver and you're visiting your friend does not get tired even though your friend is not as fit as you are or whatever. You always think your friend's, I don't know if you think your friend's more or less fit, whichever way it is. In general, if you live in Denver, then you will actually be acclimated. And the other thing you'll notice, after a couple of days in Denver, you'll be a little less out of breath. Your body will have compensated for the lower oxygen pressure. And the way it compensates is through heterotropic allosteric modulation, which is a big word for saying it makes another molecule that twists the hemoglobin into a new shape. It's actually not so much a new shape as an old shape that we've seen before, but it's a little bit surprising because it twists it more into the T state. Now you might be thinking, don't I need more oxygen? Why am I going to the T state if it binds less oxygen? Well, the reason is you need more oxygen. You need more oxygen transported. You don't need more oxygen overall. You don't need just more oxygen on the y-axis at one point. You need a bigger difference on the y-axis between lungs and tissue because the lungs pressure has dropped. Okay, this is all easier to explain with a graph than it is for me with words. So let me just explain what this molecule is that makes you high altitude tolerant and what it does, because it's actually surprisingly simple. Here's a molecule 2,3-bisphosphoglycerate. If you had to memorize glycolysis, well, number one, I'm sorry, because I don't think people should be memorizing glycolysis. I think you should be reasoning through it, because glycolysis is reasonable. But if you don't have that, if you had to memorize it, well, congratulations, you know one thing early, because you'll be able to reason out what this structure is. This is a glycolytic intermediate called 2,3-bisphosphoglycerate. Okay, and what happens, normally it's in glycolysis, your body pumps out more of it. It like pulls into the assembly line for glycolysis, or the de-assembly line, because you're breaking apart glycolysis. And you takes this molecule out of the line and pumps it into the red blood cell area because it will actually change the shape of hemoglobin. Now, it's really cool how all this came about. We're more concerned right now with how it works in the present. And so what it does is it binds hemoglobin, but it doesn't bind near the heme, it binds distant from the heme. Therefore, it is heterotropic, one molecule changes another's binding, and it's allosteric. It causes another shape to the hemoglobin, and that's how it works. This is what it does to the binding curve. So if you look at this binding curve, normally everyone has about 5 millimolar BPG in their tissues. If you have 0 millimolar BPG, your hemoglobin's actually messed up because your hemoglobin will be all the way on the black line. What happens when you move to Denver from Seattle is that you raise the concentration of BPG in the blood, And so the BPG goes up over a couple of days from 5 millimolar to 8 millimolar. The binding curve for hemoglobin goes up as well. I say it goes up. Technically, it goes down because the more BPG you have, the lower the binding curve. Now, this is where you might think it's a little bit weird because technically you are reducing hemoglobin's affinity. But the important thing is you did the foundation so you can look at this graph and you can look at the difference in the y-axis corresponding to a difference in the x-axis. The normal state is from 12, and I don't know if you can see my mouse, so I'll try not to use it too much. The normal state is on the blue line. You are talking about a difference between about 13 kilopascals and 4. That's a big difference on the x-axis, and you plot out the difference on the y-axis. And it turns out to be a 0.38 or 38% difference on the y-axis. But when you get on the plane, you step off the plane in Denver, you're still on the blue line. Your hemoglobin is still on the blue line, but the partial pressure has suddenly dropped to around 7 kilopascals. That means that you're loading up at this point. You're loading up at the upper point, but then you're dropping off at the same point because your partial pressure in the tissues hasn't changed. The inside of your body hasn't changed. It's the outside that has. And so the thing is, the y distance, the x distance has been reduced, and so has the y distance. The y distance is all the way down to 0.3. That means that your hemoglobin transport efficiency, the percentage of your hemoglobin used for oxygen transport, is now down by only 8% of the total population, but it's down from 38% to 30%, which is almost it's a 1/4 to 1/3 drop in efficiency. That's why you're gasping. It also changes the hill plot, by the way, because the N is being decreased from about 3, close to 3, to about 2.5 when you plot this out. So you have less oxygen binding, but you have so by the way, that was all. I was describing what was happening on the blue line. When you've spent a couple days in Denver, your glycolysis pumps out some more BPG, and that changes your hemoglobin. So now you have less binding overall. You're like, something's wrong here. But you're on the green line now. And look what your green line does to the oxygen transport. You're still in Denver, so you're still at 7 when you start off. You're 7 in the lungs. And you're still at 4 in the tissues. But by lowering the BPG, lowering the line to the green line, all of a sudden the difference is now back to 37%. The y-axis difference for the x-axis difference has now increased because you have decreased binding overall, but you have decreased binding in the tissues more than you have decreased binding in the lungs. Again, hard to describe with words. I want to give you the graph, and then on the test, I'm going to have you describe some aspect of this back to me with words. So you can't get away from the words, but you have to use words to show you understand the graph. This is what the foundations was leading to. So now I want you to finish the foundations, put this together with the homework, and this will allow you to explain why is it that the curve shifts down but transport increases. Oxygen transport has improved back to about where it was before, 37%. Oxygen transport is the difference on the y-axis, not the absolute value. So what does BPG do structurally? Well, we get a crystal structure of it, and we find out that, number one, it's all in the T-state conformation, and number two, BPG is right there in the middle. Remember when we go from T-state to R-state? This is arranged the same way as the other thing. Remember how the middle closes up from this perspective? Because the C-terminus moves all the way down, right? Well, the BPG goes into the middle of hemoglobin, and it prevents this from happening. So it's literally like the sabotage, okay? Sabotage comes from the word sabot, from the Dutch word for wooden shoes. And I'm not 100% sure the story is true, but it was from Star Trek 6. So I think they wouldn't put it in Star Trek 6 if it wasn't true. Historically, during the Industrial Revolution, the workers were being replaced by machines. So they'd be really mad at the machines, and they had their wooden shoes. What they would do is they would take their wooden shoes, their sabot, and they would stick them into the gears of the machine, and it would ruin the machine. It would sabotage it. BPG, in a sense, is like sabotage because it sticks into the gears of hemoglobin, and it messes it up. Although the way it messes up the hemoglobin, it messes up the hemoglobin's binding to oxygen. It turns out that it messes it up in a good way for you if you are living at higher altitudes. It allows the hemoglobin to function almost as well at the lower partial pressures in the lungs. So, there you go. You have the BPG sticking right there in the middle category, and it's literally wedging stabilizing the T state by binding it and wedging the C terminus apart, preventing, or at least inhibiting, the transition that we have to the R state. That means that overall, in the population, you will have more T state, less R state. And the interesting thing is right here they've colored these residues blue because there's a bunch of positively charged residues right where the BPG binds. And that's what makes the binding site. If you look at BPG, the BP stands for biphosphate, which means it has two negatively charged phosphates on it. The two negatively charged phosphates fit right into that hole where you have the positively charged residues around it. Now you can look at that and you can see that the hole is formed by these 6 different, technically there's 3 different residues in 2 different subunits. 6 residues that form it in the intact hemoglobin, and these are positively charged residues that line the BPG binding pocket. So the transition to the R state will close the BPG binding cavity. So if you pump in more oxygen, it will actually promote the R state, and that will decrease BPG binding. So remember how all these go together. So if you have, here's the T state right here. If you pump in, if you have like super high partial pressures of oxygen, let's say you're doing that in a test tube, it's going to promote this shape, and this shape does not have the BPG binding site. There's fewer binding sites. The BPG will bind less well. So these are all tied together. BPG has a really important role to play in modulating the binding, and you can measure this. People living at Denver will make more BPG. You can talk about all these things, and there's another way that it comes into play. Fetal hemoglobin has fewer positive residues in the middle of hemoglobin. Why would that be? If you think about it, if the fetus and the mother are at the same BPG concentration, but the fetal hemoglobin will bind less BPG. That means it's going to bind less BPG, be more in the R state, and be have more higher affinity for oxygen. If you have the mother's hemoglobin has less affinity for oxygen than the fetus's hemoglobin. That means at the placenta, where the fetus is completely underwater, the mother's hemoglobin will naturally release oxygen at the same concentrations that the fetal hemoglobin will bind it. And this is all just from a couple of mutations of positively charged residues. Now, it's a lot of connections, a lot of dominoes in this path, but that's why that's one of the goals of this chapter, for you to understand that. So that is what fetal hemoglobin has to do with high altitude sickness, because there's partial pressures. The partial pressure is lower where you have the transfer taking place. Fetal hemoglobin also has to do with the treatment for another hemoglobin-related disease. This one is a genetic disease that happens for mutations that are in hemoglobin that cause, under certain conditions, they cause the red blood cells to be in a really bad shape. To be in bad shape, and that's literal, okay? So this is the first time that we saw what might be going on with this. For a long time, there's been this disease, this sort of anemia, that is associated with people of African descent. Why would this particular disease be in these genes? It turns out there's a reason. So the first view we got of it was in 1910 when in Chicago, University of Chicago, I think, there's a doctor that was looking at the blood. They had a dental student there studying to learn to be a dentist from Grenada. And the dental student was presenting with anemia. They looked at the blood and they saw this. Now, I'm a biochemist, and even I can see that there's something really wrong with these red blood cells. There's something wrong with the shape. These look like sickles. and so they called it sickle cell anemia. What is going on with the red blood cells? And remember, RBCs are basically just bags of hemoglobin. So they didn't know this yet, but maybe with a strong suspicion could be already. Maybe something's wrong with the shape of the hemoglobin that's changing the shape of the cells. So, and by the way, so this only came about because we had the dental student who participated in the first place. So somebody who is homozygous, we've gotten it down to genes. What are the genes that are causing this? And yes, lo and behold, it's a mutation in a hemoglobin gene that changes the way the hemoglobin works. But it's in a weird place. So we found out that, remember, this is a little biology that I have to remind myself of. You have 2 copies of every gene. You get one from your mother, one from your father in most cases. If you have 2 copies of the sickle cell anemia gene, you will develop the anemia very frequently. If you're homozygous, if you have 1 unmutated copy and 1 sickle cell copy, you'll be mostly fine. So this is an example of a dominant versus recessive allele. The sickle cell anemia will be the one with the disease. And one of the hints for why does this persist in the African population? It's because if you have a heterozygous person, you can quickly find that the heterozygous mutation is associated with protection from malaria. And 1 in 12 Africans are heterozygous, which means on average 1 in 144 are going to be homozygous. So the protection from malaria is actually more important genetically than protection from sickle cell anemia. So because malaria is endemic in Africa, then you have this treatment right here. You have this is not a treatment, but you have this genetic disease that developed as a protection. Well, if we can figure this out, maybe we can help the people with the disease and also help people against malaria in other research. That will be another lecture, though. This is the idea of the misshapen red blood cells that are clearly not happy, you know? So if you look at the human population, you can see a lot of different variations in the hemoglobin that sometimes have an effect and therefore are associated with certain populations. These are very small mutations, by the way, in the big picture of things. So we can find about 300 variants of hemoglobin that seem to have some effect on physiology. The sickle cell mutant is one of these variants. So the different genes are called alleles. If you have 2 for each gene, if you have 2 of the same allele, you're homozygous. If 2 of different alleles, you're heterozygous. And it turns out that the African population has this sickle cell mutation that ends up in the population. So the results of the anemia, and this is what was obvious even in the dental student, which caused them to decide to look at his blood in the first place. The anemia in general means that you're weak, so you can't do much physical exertion, short of breath easily, and that might cause you to think maybe there's something with the hemoglobin, because that's the same kind of thing that happens when your hemoglobin is not working right when you move to Denver. You have less hemoglobin content than usual. Remember that graph where we had the anemia patient with 50% of the hemoglobin? That would be a sickle cell patient because they have less concentration, they have fewer cells, they have less hemoglobin in their blood overall, and they're at 50% concentration, which means they can transport 50% of the oxygen. No wonder that you're out of breath. Another thing that happens is that the sickle cells can actually block capillaries, which can cause the tissues to die, it can cause pain, it can cause kidney problems in particular. But here's some testimonies of what it actually feels like. And just saying that it feels like pain does not communicate how painful it is. And when you hear these patients, you've got to listen to them. It's like a jackhammer on your back, pain throughout the whole body, because when you have a sickle cell episode, you have more sickle cells than usual. They're clogging up your capillaries, and it's like torture. And that's what this is talking about. But the thing about this is that there's no obvious reason why. You can't see the capillaries. And so they go in, and sometimes they're accused of looking for drugs and making it up that it's painful. This is just exacerbated by institutional racism that causes the testimonies of pain to be misbelieved. So we've got to listen to these testimonies. And Day of Common Learning connection right here, listening to the testimonies and being open about the pain is the first step to being able to realize there's something physical going on here. And we can heal bodies, which was point number 3 of the Day of Common Learning, by looking at this. So, just look at this. This is that was a 38-year-old. An 8-year-old talks about it. You can get fevers that result from the damage being done to the body, and your eyes can turn kind of yellow. And these are the signs that she needs to go to the hospital. So this is a debilitating disease, and it's going to happen sort of without warning, sort of when the sickle cells sort of build up, or there's an episode that causes it. The main thing is a structural mutation that causes a structural problem in the hemoglobin that causes a structural problem in the cells. So if you look at the primary structure, the sickle cell anemia always associates with one little mutation. Glutamate at position 6 of the beta chain has been mutated to a valine. That's not a big difference, is it? I mean, it's a difference of charge, but there's a lot of residues in hemoglobin. Why would that one little change cause such a big deal? By the way, there's a simple way of representing this at the bottom, beta E6V. So primary structure is different, and actually that comes from one nucleotide being changed can cause that glutamate to valine mutation. But so hemoglobin A and hemoglobin S will generally, at the tertiary structure, the secondary structure is going to be the same. The tertiary structure is going to be a little bit different because you can see sort of a hydrophobic patch on the beta chains. And that means there's a sticky hydrophobic surface there. And you know hydrophobic surfaces, if you have high enough concentration, they aren't very picky. They're sticky and not picky. And so what happens with hemoglobin, you've got 2 of them. And if you've got 2 sticky ends and a high concentration of the molecule, pretty soon it doesn't take much to tilt that over to all the sticky ends coming together. Then you have a quaternary structure interaction where you have a fibril forming, not from beta structure this time, but this time from the fact that you have a dimeric and therefore symmetric protein, technically tetrameric, but it's dimeric in the sense of beta chains. And it causes it to have 2 sticky ends. They stick together and they form a fiber that will mess up your red blood cells. Not the same results as the brain-related diseases. This is a blood disease, but the same basic idea, the same biochemical problem. And then you get this. Now capillaries, here's a picture of capillaries. Some of them are so thin that they're only 1 cell wide, and this, like the red blood cell, actually has to be deformed. One of the reasons why they're so streamlined is capillaries are really small, and the red blood cells can just barely squeeze their way through. That's if they're their usual flexible selves. If they have all these spiky things inside them, then they can't fit through anymore. And then they block, and then the tissue starts to die. This shows that the kidney infarcts are often what's going to happen because the kidneys filter the blood. They have a lot of capillaries, and they have a lot of things that can be blocked, and therefore can enter into necrosis. But you can, because this is a blood thing and the blood goes everywhere, you can have strokes in the brain because of this. You can have problems in the eyes. You can have problems all over, and that's why you can have the pain all over. And it's important for future doctors to understand this, that you have this disease that doesn't have symptoms that you can see, not unless you're looking at the cells under a microscope. But so how do we see it? How can we see it short of taking blood out of the patient or short of using a microscope? Well, this is an interesting way to see it. If you actually hit it with a photoacoustic microscope, you hit it with sound waves, it will actually resonate differently, like it's a drum that's too tight. And this will actually cause it to be, this has a big effect. If you hit it with sound waves, it will resonate with the sound waves differently. And it will sound different to this special photoacoustic microscope. Maybe someday we can have some kind of advanced device that can somehow detect this, maybe even through the skin. For right now, it's sort of a proof of concept, but it shows that the red blood cells are very different with the way that they act, the way that they are shaped. Their morphology is the word. Here's another diagnostic that we can do today that's very simple. Because the red blood cells are messed up and they're all tense, they actually will sink faster. They're more solid, and they will be, all you need is a centrifuge. And the thing is, you can make a centrifuge pretty cheaply. All you need is an egg beater, or you need a little spinner, a fidget spinner can be turned into a centrifuge. So if you can have a cheap centrifuge, you don't even need electricity necessarily to run this particular assay. You put your blood in the tube, you have some other stuff in the tube to make it sort of thick so that the blood will have something to pass through, a little bit gel-like. And it turns out that if you separate the blood, the red blood cells will go down halfway down the tube, but the dense red blood cells, which will be sickle cells, will go all the way to the bottom. And so you can actually detect the sickle cells directly just by looking at it using this. No electricity, you just need to have the little tubes which you can carry in a backpack, along with a hand-operated centrifuge. Very, very cool stuff. And so, yeah, here you have an example of, you have a genotype of the homozygous for non-hemoglobin S, and you can detect, you can have the sickle cell hemoglobin that will show up in the tube at the bottom. You have a big red band at the bottom because they have the sickle cell anemia. They have so much of the S hemoglobin that it's forming fibrils. And this is actually a big deal, especially having been in Burundi myself and seeing the challenges that they face to getting this medical care. Is something like that can actually really save lives. So just remember, all of this comes from a single point mutation that is very small. So I have a couple more things. These are more things I want you to understand, not memorize for the test necessarily, but these are really cool advances. You need to know in general how these treatments work, okay? So there's treatments for small molecules. This is a big change. Fibrils are very big and they're very hard to get apart. But still, maybe we can come up with a drug that blocks the sticky patch. And there are some people that are trying to do that. They come up with this drug, voxelotor, or however you say it, and it actually is approved for sickle cell disease patients. This is a drug company that made it. And yeah, I don't want to say too much about it, just that one company at least is working on this. But I wouldn't be surprised if it didn't work that great, because you've got one little drug, you've got this big interface, and you've got this really tight interaction. Maybe there's a limit to what we can do with drugs, but there are other ways we can go after this. So the other thing that we can ask is, why does sickle cell persist in Africa? And it looks like it persists for a reason that is around today. This is not just some kind of paleo-ancestral thing, which sometimes happens when you have these things like lactose intolerance, for example. But you have an actual current reason for this, because babies are still being born with sickle cell anemia with the HBS gene. And there are reasons, there's got to be some reason why it's protective from malaria. So we want to know, why is it protective from malaria? Here's the geographical association of the gene with malaria prevalence. And you can plot out malaria prevalence in yellow, and you can show where the gene is in the colors on the left side. And it turns out that, right in here, it's the same number. 8% of the African population carries at least one gene. So this made people say, I think that there's got to be, and malaria lives in red blood cells. So it seems to make reason that messing with your red blood cells a little bit might keep the malaria out. The problem is, when you mess with them a lot, you have other problems. You have the sickle cell problem. And so this was a hypothesis. Now we have got it down to a science where I am absolutely certain I can replace that question mark with an exclamation point. That's only happened in the last 10 years. Here's one paper that does about it. I'm giving you the titles of these papers so you can look into it yourself for another class. Believe me, we have enough to read right now in this class. But here are the papers. Sickle hemoglobin specifically confers tolerance to malaria infection, Plasmodium. In fact, they found that specifically hemoglobins S and C actually interfere with actin remodeling in the red blood cells. So they were seeing that it messes with the red blood cells specifically by messing with actin remodeling, which has to do with how the cells move and shift around. The sickle cell fibers interfere with this process, and there are trafficking networks in the cell that bring the proteins to the surface. You've got to move through the cell. So maybe this is kind of preventing the proteins, the malaria parasite proteins, from moving through the cell. You're kind of impeding them, putting a roadblock in their way. And this just came out. It's actually the physical tension of the red blood cells that prevents the parasite from invading as well. So it's not just trafficking, but it's also invading. And this is a really cool study that just came out about exactly how this works. Hopefully, by understanding exactly how the disease is prevented, maybe we can figure out how to replicate that without causing the disease. Without, the disease malaria, we can stop malaria without causing sickle cell disease. And selection for this trait is still happening. We're seeing that hemoglobin S is still higher because malaria is still endemic. But maybe you've heard some good news on this front. This just came out. The World Health Organization has approved a malaria vaccine. I have never been able to say that before. It's actually, so there's hope that the mRNA vaccines might be applied to malaria, but there's not been time for that to develop yet. This is a typical old regular protein vaccine, but finding the right protein to give in the right way, in the right combination to alert the immune system has been really hard. Malaria is a total pain to deal with. Parasites have all these tricks where they are just trying to get away from you. But it looks like this is working better than anyone has before. Notice it reduces severe malaria by 30%. Compare that to COVID vaccination, which reduces severe COVID by like 95%. This is why I am still very happy with COVID vaccines, even though it might have dropped to 80%. Dropping to 80% is not a big deal when you have a really bad disease. I mean, it is a big deal that we need to do better, but it still means the vaccine is working really well. So don't get me wrong. The severe malaria cases, by 30%, is good enough to be worth a 4-injection thing. So you think 2 injections for COVID are bad? Well, talk to the hand, because malaria has to deal with far worse for a far more complex disease. COVID is definitely, let's not put diseases against each other. COVID and malaria are both really bad, but we've got to recognize the specific malaria challenges that we face. So all that is to say, I'm really glad to be able to pass this on. This is why I wanted to spend some time on this so that you would know about this. And then also we have this is where what we need to do is we need to get vaccinations and treatments into places like this. This is what Burundi was like. This is the hospital at Burundi. This is actually in the Democratic Republic of Congo, which is right next door to Burundi. This is what it looks like, and this is where we need to help people. So we need to bring this knowledge, we need to bring our efforts into this place, because these people must come first when we're talking about eradicating malaria and curing sickle cell disease. So with that in mind, how can we cure sickle cell anemia? Well, one of the ways is with this little molecule, which is weird. It's hydroxyurea. If you remember the structure of urea, it's urea with a hydroxy on it. But I don't know how they found this out. But this actually will, if you give this to somebody, they will actually make more fetal hemoglobin. So we've talked about fetal hemoglobin. Think about how it works. It has a higher affinity for oxygen. So if you stimulate more fetal hemoglobin, your body will be able to bind more oxygen. And usually you turn fetal hemoglobin off, but maybe we can find a way to flip that switch and flip it back on. Hydroxyurea does it somewhat, doesn't do it very well, but does it enough to help some people. The other thing you might say, we get into the genes. The genes had the mutation in the first place. It's just one nucleotide, right? What if we could edit that nucleotide? Now, it's not that simple because you got to edit it in all the red blood cells and you got to make it stick. You got to make sure you're not editing anything else. But this is part of a therapy right now. And we are going to talk about CRISPR and its complications, its DNA metabolism. So we're going to talk about that in Chem 4363. Okay? So take that class if you want to talk more about CRISPR. And just know that CRISPR gene editing is actually in progress right now. Here's a researcher who has focused her life because she has this disease herself. And her sister has it more severely than she does. Like we said, like any disease, this has more severe and less severe manifestations. She's from Liberia, and she is working on treatments to encourage the production of fetal hemoglobin. So something like hydroxyurea, but something that we understand how it works, and we can make it work better. Her current goal is to develop a molecule. Normal levels of fetal hemoglobin are around 1%. If we can bring that up to 15%, that could have a real effect on how the blood works. In fact, she might be able to get up to 30%. So let's root her on, and let's see what comes out of her lab. By the way, this all has to do with a transcription factor that silences the fetal hemoglobin. So what she's trying to do is mess up the transcription factor, possibly with an antibody fragment, which brings it back to the antibodies that we've talked about. All this is to say a lot of details, but we just want you to know what is her strategy. And it has to do with fetal hemoglobin as well. Here's just a little taster about what you can do with gene editing with CRISPR. You have the incorrect base that it is a T in the beta globin gene. You can replace it with an A, and then you have normal red blood cells. All the details about the Cas9 and how it works and all that stuff will come later. I just want you to notice that you can edit the beta globin gene, or maybe you can edit the fetal hemoglobin gene. That's what they have on the right here. So if you can edit genes to maybe remove the repressor so that then you don't have your fetal hemoglobin turned off, you can flip that switch on that way. So there's a lot of levels to talk about here. Here's a really long, good article from the New York Times that I will post on Canvas. This is FYI, which means I don't expect you to read it, but maybe I want you to read it when you have time. So I encourage you to read it and maybe use it for another class. And maybe we'll be able to talk about this like in capstone sometime or something like that. Because this involves the politics of the presidential administration committing the spending that will speed the clinical trial results, which are very promising right now. There are people, and you can read about her story right there. Another person talking about this, so this is gene therapy. And so Francis Collins, who was the head of the NIH, in fact, he is the head of the NIH until the end of this year. He's finally stepping down, but he's actually involved. He started the organization BioLogos that I am involved with, which deals with questions of science and faith. And he actually gave a talk about the joyful complementarity of science and faith. I encourage you, if you need to watch a science and faith talk, you can watch the whole talk. If you just want to know what he says about what the state was just 2 years ago of gene therapy for SCA, you can watch the thing I have there. I really do think he is an MD. He talks a little bit about medicine. He talks about science and medicine. And he is a Christian who talks about how his faith fits with the whole view of science from evolution, but it also motivates him to heal people in a way that is constructive and is truly building. He is giving glory to God by doing science. And so, again, I encourage you to look at that for the science and faith angle rather than the science and politics angle. But he's also, of course, involved in politics. And I'm looking forward to seeing what he's going to do now that he's finally stepping down from the NIH. He has been the head of that for so long, and in my opinion, he's done a really good job. Because you have no idea how hard that job is. I have no idea how hard that job is. Okay, so that's why I wanted to add this so I could go on a little bit about the important parts. But the most important, the looming problem, the proximal problem that you have is what is the test going to be like, which is going to be on Wednesday. And so finish up the foundations work that we have. And we have the achieve problems. Now that you can do all the problems on achieve, we've talked about all of them. So work your way through them. Look at the extended solutions when you need to. And get those done sooner rather than later. So if you have questions about them, email me before class on Monday. If I get questions that I can answer to everyone, guess what? I'll explain it to everyone. It's a really great deal. So I can tell you what the answer is. Or I can tell you that thing's not important for the test. That's the other thing. All right. So if I haven't gotten you this weekend, there will be some work to ramp up to the test. We'll review on Monday. We'll take the test on Wednesday. And then biochemistry will have some more days off coming off very soon. So get over this last little hill, and then we'll have more tests as we go along. All right. Thanks for listening. I will see you on Monday.